Have you ever poured your heart and soul into an experiment only to get puzzling results? Life scientists will experience it multiple times. It is natural that the more ambitious the scientists are, the more often they would experience puzzling results. We have been talking about a puzzling case in a real scientific experiment. The experimenters have already tested 3 ideas for solving the problem. In today's episode, we will discuss their 4th and final idea based on the previous 3. We will see how the idea was refined and updated when the experimenters identified what were unknown in the experiment. This updated idea was the basis for designing the actual troubleshooting experiment, and we will discuss it in the next episode. Hi everyone, welcome to the Foundational Skills in Life Sciences podcast. I'm your host, Dr. Synaptologica. We discuss the skills that life science scholars learn, use, and improve throughout professional activities. Your benefit is expressed in my motto, learn the skills, enjoy academic life. Last episodes. We are in a long mini-series of reading a life science paper. Today, we are in episode 23. If you are a new listener of my episodes, please start listening from episode 7. It was the start of the mini-series of reading. We are now discussing a real story. It is a story about one specific experiment that did not work well at first. The experiment is not specifically related to our milestone article 1 by the Nobel Laureates. But we are discussing the story because it will tell us that negative controls are important when we design life science experiments. It will also tell us that negative controls are important when we detect potential problems in experiments and when we troubleshoot them, always together with the positive control counterpart. In the episodes 20 and 22, we talked about how the experimenters performed 3 troubleshooting experiments based on 3 ideas. Here's a brief summary. The idea 1 said that the cell function could have been wrongly increased during the chemical treatment period when the cells were treated with any chemical compound of interest, irrespective of whether the compound was active, inactive, or irrelevant. Idea 1 was shown to be incorrect by the first troubleshooting experiment. It included a new negative control where no compound was added during the treatment period. This was the no treatment negative control 1. The idea 2 said that the cell function could have been wrongly increased while the cells were simply kept for a long 1 hour during the treatment period, even without any chemical treatment. Idea 2 was shown to be incorrect too by the second troubleshooting experiment. It included a new negative control where the cell function was measured immediately after the stabilization period that preceded the treatment period, without any chemical compound of interest. This was the no treatment negative control 2. The results of these 2 troubleshooting experiments indicate that there was no problem with the treatment period. The idea 3 said that the cell function could have been wrongly increased even before the treatment period, that is, during the stabilization period. This is the period when the cells were simply placed in a fresh culture medium for 1 hour. Indeed, the idea 3 was shown to be correct by the third troubleshooting experiment. It included a new negative control. The cell function was measured immediately after the culture period, still in the old culture medium, and without stabilization period and without treatment period. This was the no treatment negative control 3. So, through these troubleshooting experiments, the experimenters have found that the culprit was the stabilization period. Again, the stabilization period is the period after the culture period, when the cells were simply placed in a fresh culture medium for 1 hour. Since the last episode, I hope you have thought about what could have gone wrong with the stabilization period. Let's see how your ideas fit with the ideas of the experimenters. Today's episode. In today's episode, episode 23, we will talk about the last idea, idea 4. It led to the fourth and final troubleshooting experiment that helped the experimenters solve the problem. When I prepared the content for this experiment, it became very long. This is reasonable because it is the most important and the most complex experiment. So we will spend a few episodes discussing it. Idea 4, an idea that would help understanding the problematic results. As we have reviewed just now, the stabilization period seemed to be the suspected cause of the problems. Then, what was wrong with the stabilization period? The experimenters carefully thought about it, and they came up with an idea or a hypothesis that would help them understand all the problematic results. Let's call it the idea 4, because it serves for the fourth troubleshooting experiment. Here's the idea 4 in a single sentence. Replacing the old culture medium with the fresh culture medium led to a sustained maximal increase in cell function. Let me say it again. Replacing the old culture medium with the fresh culture medium led to a sustained maximal increase in cell function. This idea can be broken into 3 parts. Part 1 of the idea 4. When the old culture medium was replaced with the fresh culture medium, the cell function was increased. So the medium replacement, or the medium exchange, was the trigger for the cell function to increase. Part 2 of the idea 4. When the cell function was increased, the increase lasted long during the experiment. And part 3 of the idea 4. When the cell function was increased, it was increased maximally. If this idea 4 is true, then it can explain the 3 strange and problematic behaviors of the cells in the original pilot experiment. The first strange behavior of the cells was that the cell function was low in the old culture medium. This was found in the no treatment negative control 3. If the idea 4 is true, this behavior becomes reasonable because the cell function was measured before the medium replacement, that is, immediately after the culture period, but before the old culture medium was replaced with the fresh culture medium. Then this low function can be considered as the new baseline level in the absence of any stimulation. The second strange behavior of the cells was that the cell functions were high in both the positive control and the mock treatment negative control. If the idea 4 is true, this behavior becomes reasonable because the cell function was measured after the medium replacement. The function was increased and maintained high through the later part of the experiment, that is, after the stabilization and treatment periods, and also at the time when the cell function was measured. The third strange behavior of the cells was this, and this is a key point. Not only the cell functions were high in both the positive control and the mock treatment negative control, but also their functions were high at a similar level. If the idea 4 is true, this behavior becomes reasonable because the cell functions in those groups had been maximally increased in the same way before the chemical treatments. Then the chemical treatments would not matter. In other words, the chemical compounds would not change the function further because A. Treatment with an active compound would not increase the function because the function was already increased maximally, reaching a saturated level. There was no room for further increase. This situation corresponded to the positive control of the original experiment. And B. Treatment with an inactive compound would not change the already increased function either. This situation corresponded to the mock treatment negative control of the original experiment. Now you see that if the idea 4 holds true, everything in the original problematic pilot experiment can be explained logically. Everything fits together perfectly. 4 unknown things about the idea 4. The intention of having a stabilization period was that the cell function would calm down and stabilize during the stabilization period, because the experimenters did not intentionally stimulate the cells during this time. The calmed down, stabilized state would make it easy and clear to detect an increase in cell function by an active compound later. But the experiment would not work well if the stabilization period actually stimulated the cells instead of calming them down. It was good that the idea 4 could explain all the strange behaviors of the cells. It indicates that something strange could have happened. Then the experimenters carefully analyzed the idea 4. They noticed that there are at least 4 unknown things about the idea. The experimenters wanted to isolate the unknown things and make them clear so that those things can be addressed separately in the troubleshooting experiment to follow. The first 3 unknowns correspond to the 3 parts of the idea that we have already talked about. The 4th unknown is new in our discussion. Unknown 1. The first unknown corresponds to part 1. It is at which time point of the stabilization period the cell function was increased. Was it at the start of the stabilization period when the old culture medium was replaced with the fresh medium? Or any time later in the 1-hour stabilization period? Unknown 2. The second unknown corresponds to part 2. It is whether the functional increase lasted long during the experiment. In other words, practically, did the functional increase last up to the end of the treatment period when the cell function was measured? Unknown 3. The third unknown corresponds to part 3. It is whether the functional increase was maximal in degree. In other words, did the function reach a saturated level so that it could not be increased further with a stronger stimulus? And unknown 4. The 4th unknown is new. It is what caused the cell function to increase directly. In other words, what happened when the culture media was changed from the old medium to the fresh one? What the experimenters realized. When the experimenters reached this stage of thinking, they carefully examined all procedures of the experimental protocol and the compositions of all media used in the experiments. Then they realized one important fact. During the culture period, the cells were bathed in the culture medium that contained animal serum. After the culture period, the experimenters replaced the old culture medium with the fresh one. We have been talking about this media exchange repeatedly. But what they realized at this stage was that this fresh culture medium did not contain any serum. This means that when the old culture medium was replaced with the fresh medium at the start of the stabilization period, the serum was removed from the medium accidentally. Professor's viewpoint. Here, please let me give you a brief comment. I will call it a professor's viewpoint. It is a personal comment, but I think it is an important comment. I just said that the experimenters realized that the serum was removed accidentally when they changed the old medium to the fresh one. Some people might think that this information should have been apparent to them from the very start. They had access to the information about the compositions of all media. The experimenters were doing the cell cultures for a long time, and they did not change the compositions of the culture media. And at first, the functional measurement was new to them, but later, the experimenters did the experiments many times by themselves. So it might seem strange that the experimenters did not pay attention to this fact, that changing their media would remove the serum. They were completely unaware when they designed the original pilot experiment and while they were doing the troubleshooting experiments up to this point. But please let me add that this situation happens frequently. Once an experimenter believes that a protocol should work fine, it is often challenging to question it and revise the thinking. When we troubleshoot, it is necessary to review many aspects of what we have been doing. They include the overall protocol, the individual reagents, and the practical details of all the used methods. There might be many more. We would like to reevaluate as many things as possible, carefully and objectively, from a fresh viewpoint. This needs flexible thoughts and a healthy dose of skepticism. I believe this is very important, but this is often very difficult. But in our story, the experimenters could identify that there was something strange with their protocol. In this case, it was the composition of a cell medium that they have been preparing all the time. I think that the experimenter's attitude is very commendable. Now back to the main content. Updated idea 4. So the experimenters became aware of this new information. The serum was removed when the old culture medium was changed to the fresh one. Based on this information, they refined and updated their idea 4. The original idea 4 stated that replacing the old culture medium with the fresh culture medium led to a sustained maximal increase in cell function. The updated idea was more specific about when the function was increased and what caused it. The updated idea 4 states that removing the serum from the culture medium immediately led to a sustained maximal increase in cell function. This updated idea 4 formed the basis of how they designed the 4th troubleshooting experiment. We will discuss the actual design in the next episode. But before talking about the actual design, we would like to interpret the updated idea a little further here. The issues that the updated idea 4 addresses. In the 4th troubleshooting experiment, the experimenters would examine the effects of removing serum from the culture medium on the specific cell function that they were measuring. More specifically, they wanted to examine whether the serum removal changes the specific cell function, and if so, when, for how long, and to what extent the function changes. Corresponding to this statement, the updated idea 4 has 3 parts, similarly as the original idea 4. The experimenters wanted to test if all 3 parts of the updated idea 4 are true. All 4 unknowns in the original idea 4 are already incorporated in the updated version. Here are the 3 parts of the updated idea 4 and the individual comments about how to test them. Part 1 of the updated idea 4. When the serum was removed from the culture medium, the cell function was increased immediately. This part 1 addresses the unknown 4 in the original idea 4. What caused the cell function to increase directly? The causative mechanism was specifically defined as the serum removal from the culture media. This part 1 also addresses the unknown 1 in the original idea 4. At which time point of the stabilization period was the cell function increased? It will be immediately after the culture media were changed at the start of the stabilization period. By the way, to generalize our discussion, I'm using a very vague expression, immediately. Immediately could mean milliseconds to some of you, but this number will not be important for our discussion. If you want to obtain a rough idea here, it would be okay for me to say that it takes several minutes for the cell function to increase. But it is also okay to say 5 to 10 minutes. Here's a quick comment. Exactly how long it takes for the cell function to increase will depend on multiple parameters. They would include 1. How quickly the media are exchanged. 2. How quickly the serum concentration is reduced to 0 just outside the plasma membrane of the cells. 3. How quickly the serum removal is converted to intracellular signals inside the cells. And 4. how quickly those signals affect the specific function that the experimenters were measuring. Now, back to the main content. Part 2 of the updated idea 4. When the cell function was increased, the increase lasted long during the experiment. This part 2 addresses another unknown, that is, the unknown 2 in the original idea 4. Does the functional increase last long during the experiment? Practically, it can be expressed as, will the functional increase last at least up to the end of the treatment period when the cell function was measured? To test if parts 1 and 2 are true, the experimenters plan to measure the cell function before and after removing the serum. This way, they could directly record the time course of the functional changes. We can call it a no-treatment negative control 4. This naming, using the term negative, becomes a little tricky here. On the one hand, it is a no-treatment negative control because this group is not treated with the active form of the chemical compound that was reported by other researchers to stimulate the cells. But on the other hand, it is not a strict negative control because the idea 4 assumes that the cells could be stimulated by serum removal. For simplicity here, we will still call it a no-treatment negative control. Part 3 of the updated idea 4. When the cell function was increased, it was increased maximally. This part 3 addresses still another unknown, that is the unknown 3 in the original idea 4. Was the functional increase maximal in degree? The updated idea 4 says, yes, it will be maximal. To test if part 3 is true, they would examine to what extent the cell function can be increased. For this purpose, they planned to create a new positive control that is slightly different from the positive control in the original pilot experiment. In the newly added positive control, the cells would be treated with the same active compound as in the original positive control, but at a higher concentration than the original concentration. The original concentration was the one that was reported by other research groups to give the maximal stimulation. So the cells in the two positive controls will be treated with the active compound at the maximal and supramaximal concentrations. How to proceed further? We have seen that the ideas of these experimenters are systematic and logical. I'm very much impressed by that. Do you now think that the experimenters are fully ready to start the fourth and final troubleshooting experiment based on the updated idea 4? The fact is that they were not there yet. There was one more challenge to the story. Without understanding this crucial fact, or a missing piece, the experimenters would not be able to design the experiment properly. So they would not be able to conclude that the serum removal was the direct trigger of the cell stimulation. What was the missing piece of information? How would you proceed if you were a team member? I just mentioned a missing piece of information, but in fact, I have already provided you with the necessary information. The information is already there. I already mentioned it as a fact, but I did not mention it as a crucial fact. So the information is not missing, it is in a way hidden from a naive mind. Can you think of what it can be? Which crucial fact do we need to consider for designing an effective experiment? This is it for today. In the next episode, we will talk about the crucial fact, how the experimenters designed the fourth and final troubleshooting experiment based on the crucial fact, and what they found. Your action. Until we meet next time, think about what the crucial missing fact might be for designing the experiment. Do you want a hint? I would be happy to give it to you. Pay attention to how to change the media practically. When you get an idea, please send me an email about it. We can think about it together. My email address is ideas@synaptologica.com. Thank you for joining me in today's episode of Foundational Skills in Life Sciences. I'm Dr. Synaptologica. I'll see you soon. Bye for now.